on our trajectory through the brain, and also how we can try to understand brains at the system level and exploit that for technology. And as part of that ambition, we're extremely happy that we have Eberhard Fetz with us today of the University of Washington. Eberhard started life as a physicist, but as you all know, this is not directly of any positive or negative consequence for people's future. But what Eberhard, then on the basis of this done, he looked at the brain not necessarily as a physiologist or as an anatomist. Okay, how is this structured, or what are the response properties of the system? Eberhard has really gone in the direction, okay, but how can we understand the brain also by interfacing it to external devices? And so for many years now, Eberhard is really leading the field in this whole domain of brain-machine interfaces. But what is really special there, which I really appreciate in Eb's work very much, which I think is really outstanding, is that it's not just an engineering exercise, like, oh, let's just hook these systems together and something will come out, and then you write an exciting paper about it in Science or Nature, like, oh, for the first time we managed to control a bicycle with the brain. But what Eberhard has been doing, which I think is really very powerful, is it really advance an agenda of understanding how brains really encode and deal with the world. And that means an engineering component of neuroscience is really also, let's say, a window into brain organization and brain function. That makes, I think, his contributions really important and outstanding. So it's really great that you're here.
Well, thank you very much.
Thank you very much for joining us, and looking forward to your talk.
Yeah. Well, thank you, Paul. You can go on with this wonderful introduction, but I guess I should talk. I appreciate it. Thank you. And I actually want to start by thanking Paul and Anna for allowing me to participate in this wonderful meeting in this wonderful city, and tell you a little bit about something I think is relevant to the theme of the meeting, which has to do with bidirectional interactions between the brain and implantable computers. So this title would be totally unremarkable without the word implantable in it. So because you know that our brains interact bidirectionally with computers all the time, because they're two sort of complementary information processing devices. The brain finds a lot of interesting interactions between computers, and technology has now gotten to the point where fairly powerful computers are small enough to carry around in your pocket, and you can see a lot of interactions between these things and brains on buses and so forth. But this is probably close to as small as a computer is going to get and still be able to interact with our brains through normal sensory channels, that is vision and touch and voice even, and audio. But except maybe the Google Glass will be the next step to make it even smaller. So the question is, how small can these get? Technology is actually making these computers, the computing part of it, small enough to implant. But the question is, what happens when you have more direct connections between a computer and the brain? And that's basically what the theme of this talk is. So by that I mean a direct connection from cells that are being recorded in the brain and triggering stimuli contingent on that activity back into the brain. So this is what I call a recurrent brain-computer interface, or bidirectional brain-computer interface, that we've explored with monkeys carrying these around. So basically, there are two types of results of having this artificial recurrent connection to the brain. One is that the brain could actually learn to incorporate that artificial loop into normal behavior. And the second is that spike-triggered stimulation generates synaptic plasticity, can strengthen and weaken connections. And I'm going to show you examples of all of this. So this recurrent loop is sort of a variant of the more traditional brain-machine or brain-computer interfaces, in which neural activity is taken, tapped from the brain, usually in sites in motor cortex where cells are already used to having very variable activations appropriate for activating biological muscles, but taking those signals and operating external devices. So now we can close the loop and see what happens when, instead of external devices, we have these cells stimulate the brain. So this paradigm obviously requires that the brain learn to optimize the control of these signals. So there's what I like to think of as a volitional self inside the brain that operates these devices. And this self can learn to control external devices. There's Ripley in a power gripper in the Alien movie. And I like to think of this as actually a metaphor for the self in the brain controlling the body. And we know that our self in the brain can learn a lot of new paradigms of control very quickly. And I want to show you a couple examples. So here's this wonderful study of Shadmir and Musa-Evaldi, where human subjects were controlling the position of this, I guess, gimbaled arm that could be moved from center position to different targets. And it was connected to a servo motor that could induce sort of perverse force fields that were dependent, in this particular example, on the X and the Y velocity of the hand, something the brain had never experienced in real life, but adapted to relatively quickly. So the first couple trajectories with the servo motor turned on were deflected in the way shown here. After a couple hundred trials, soon the subjects were making fairly direct movements. And in fact, the brain had readjusted itself to this environment, as proven by the fact that when you turn the motor off in catch trials, the trajectories were now deviating in the wrong direction. They were right for this field, but wrong for this field being off. So we have this miraculous device inside our heads that can do this sort of adaptation fairly quickly. And that bodes well, actually, for control of brain-machine interfaces. I want to show you one more example. This is from the lab of Andrew Jackson, who's a former colleague. And basically the idea was to see how humans could learn to contract their muscles in new combinations. So there was a cursor that as a human subject with the arm sort of fixed, and the cursor had to go into the target. And the cursor was the sum of vectors produced by 6 muscles. So for each muscle, it was contributing a vector contribution in its assigned direction with a magnitude that was proportional to the amount of activity in that muscle. So these directions could be assigned in a relatively intuitive way, so that the flexor muscle would move the cursor in the flexion direction. And subjects very quickly learned to get the cursor into the target. But the directions could also be assigned in a non-intuitive way. So the flexor muscle would now move the cursor in another direction. And initially, this resulted in very weird trajectories. But over only a couple hundred trials, these trajectories got more direct, and the time to acquire the targets was much less. So this is the adaptation of the brain to this new set of, let's say, directions that the muscles were moving the cursor. And this is all done intuitively. I mean, this is all taken care of by this wonderful machine that we have. And a similar sort of thing actually now has been shown for motor cortex cells. The reason I'm showing this is the same sort of thing can be demonstrated for cells in motor cortex that are linked to the direction of a cursor. This is Jose Carmina and Granguli. Maybe you know this, but it's a very similar sort of a paradigm. So we have this ability to control the activity, or learn to control activity in a natural way that controls an external device, even in ways that these cells don't normally operate. So what is the evidence for the degree of control of these cells? I pose that question as an excuse to go back to experiments that I did a long time ago as a postdoc, in which the experiment was to see how much a monkey could control motor cortex cells with biofeedback. And the idea was, for example, to see how much the monkey can control the firing rate of a motor cortex cell. And it was implemented by having pulses triggered from the action potentials of the cell and doing a sort of leaky integrator of this firing rate. When it reached a threshold, the feeder discharged. Here's the meter. There's feedback here continuously on the level of this activity in the form of a meter. Too bad I can't show you the rest of the picture. It's taboo to show illustrations of primates for recorded talks. So you have to imagine the monkey sitting here watching this meter. And basically, the monkey very quickly learned that when this meter deflected over here to the extreme right, it corresponded to this threshold, and he got his applesauce, which went through a tube. And it took the monkey not too many sessions to learn this paradigm and to then be able to control newly isolated cells in relatively quickly. So the cells were recorded through this microdrive. And so here's an example of a session in which the firing rate of a cell is shown in 1 minute averages. And then the reinforcement period is begun, the meter's turned on, applesauce is available for these high rates, and the monkey is successively increasing the firing rate to much higher levels during this reinforcement period. He does it by increasing, well, obviously we were tweaking the threshold all the time to keep pushing him to higher rates. And what he did was increase the firing rate by increasing the number of bursts and the intensity of the bursts and even the interburst activity. And then the next period, the extinction period, the applesauce and the meter turned off and the firing rates drop again. So this is really volitional control of this cell activity. Now, an interesting demonstration of the degree of specificity of this control is shown in this session, in which there were actually two cells, large and a small unit, that were separated on the basis of their action potential shapes. And here's the firing rates, 1 minute firing rates of the large unit in red and the small unit. And here's the preconditioning level. And then in this session, in this part of the session, the pulses from the small unit drove the meter toward that reinforcement level, and pulses from the large unit drove it in the other direction. And what the monkey chose to do is to increase the firing rate of the small cell relatively quickly. Never seen these cells before. And relatively quickly he was increasing the firing rate of the small cell. Didn't decrease this, didn't have to actually. Didn't push it, let's say. And then during timeout, the firing rates dropped again to these average timeout levels that are shown by these horizontal lines. And then the polarity of the pulses was switched. So now the large unit drove the meter to reinforcement, and that monkey increased the large unit activity, and then during timeout it dropped again. So this shows differential control of two neighboring cells. By the way, these two neighboring cells had very similar input from the periphery. They were both involved in knee extension for some reason, but this demonstrates independent volitional control of the cell from the volitional self or whatever. When you require that control to be demonstrated with this sort of a paradigm.
Sorry, can I ask you an idea?
Yes, please.
Even though this is very surprising to me, I didn't know if this worked, but is the monkey, you think, activating populations of neurons?
Absolutely, absolutely.
Oh,
that's
so
right.
It's not just single.
Yeah. No, that's exactly right. And so this unit is part of a larger pattern, including muscles. I'll show you in a minute. Obviously, the fact that the reinforcement was contingent on the activity of a cell doesn't mean that it couldn't just be that cell. There has to be inputs that are driving that cell, and it's part of a larger pattern. That's right. So there's a whole story there's a whole issue, I should say, about what the other cells are doing and so forth, and how they can be dissociated and so forth.
What is the experience of the monkey is also interesting. If you have a human in this situation, I wonder all the other neurons are also perceived.
Well, you're right. It's a good question what they're perceiving. We were actually interested in this as a technique for identifying what they were doing, what they were going to do with particular cells. So the rationale was that this was a way of mapping the movements that the cells, a given cell was related to. So this was sort of the motor analog of receptive fields. You now get the monkey to activate the cell, find out what he does, and say this is what the cell is doing. Unfortunately, that agenda sort of went by the wayside because what the monkey was doing was quite variable quite often. It could start as being variable, it could get more specific, that was interesting. And in some cases, he was activating motor cortex cells without any movement at all. And so as a reliable way of investigating the motor map, this didn't turn out to be that great.
That's my question is, if you don't allow any feedback from the periphery and if you don't allow the brain cells to control actual movement, does he still learn it? Because you can imagine that he's thinking, I switch my knee and I'll get juice.
Yeah, that's quite likely is the initial strategy of the monkey is to find out what movements he was going to get rewarded for in that particular session. But like I say, in some cases, he started right off looking at the meter, driving the cell up, absolutely still, no muscle activity. Each cell is probably involved in a different type of circuit.
We have two questions. On the one, what's interesting is that in these intervening periods, it immediately drops back to a baseline.
So in these timeout periods?
Yeah, so
it
looks like
meter is turned off, yeah. So
no,
he doesn't see a meter.
No, wait, but so are you interpreting this with respect to the sensory drive on this neuron?
I'm interpreting it as a background level of activation of the cell. It's whatever the cells are doing. These are typically spontaneously active, even without movements. So this would be the level of activity, spontaneous activity.
But it also means that you need to maintain this reward contingency in order to drive up this activity and control the response.
Exactly, that's correct, yes, because if you stop this reward, it drops.
Exactly.
And here too. So it's a volitional control.
It's a transient state that's being built up.
Yes, yeah. It's a transient cooperation of the monkey to activate those cells.
So why do you call it volitional?
Because it's centrally originating, that's what I mean by that. It's generated by the brain, right?
Okay, but volitional would mean that there is really a conscious decision behind it.
I'm not just talking about consciousness here. I'm just talking about volitional movements, volitional activation without any implication of whether the monkey is aware of what he's aware of, we just don't know. Okay. All right, so here the first lesson is that you can independently control the firing rate of two neighboring cells right next to each other. But you notice that he doesn't actually decrease this activity when it could have been decreased. So to press this point in this session, in this next part of this session, the pulses from the large unit made the meter drift away from the reinforcement level. And if you just shut the cell down, then there was a multi-vibrator that made it drift toward the level, toward reinforcement. And what the monkey initially did is increase the rate, which is what paid off here. But then after a couple trials, he was actually suppressing the firing rate of the cell, allowing the multi-vibrator to reach the reinforcement level, and continued to do that again until the end of that contingency, and he went back to timeout levels here. So the second point here is that you can actually control cell activity bi-directionally if you're rewarded.
Sorry, another question.
Yeah. Mm-hmm.
Do you have to search hard to find trainable neurons, or is it
No.
a high rate of success at almost
It's
any neuron?
100% rate of success in motor cortex. I would say that pretty much every cell that we found, once the monkey had gone through a couple sessions to get the hang of this task, then pretty much every newly isolated cell he could demonstrate volitional control. But, yeah. So that's in motor cortex. And then in sensory cortex, let me just get off the track. Yeah. So the question is, how many areas can you demonstrate this volitional input? And I'm guessing that there are quite a few, including postcentral sensory cortex. So here's the paradigm then. We've got this reinforced activity. This is the basic biofeedback paradigm that was implemented in a variety of ways. The idea being that there's some activity that's reinforced through feedback to this volitional controller, which is the rest of the brain, obviously. And for monkeys, it's motivated by reward to motivate the volitional controller to control this reinforced activity. All right, but the elephant in the room here is this correlated activity, which has some potential interactions with the reinforced activity. By that, we mean, first of all, other cells, and also in the motor cortex, other muscles. And these interactions could be from the periphery, from this region to this, or vice versa. And this activity could be either, or these relationships, I should say, could be either adventitious, they happen by chance, or they could be necessary, they could be causal. And so we sort of explored this with the muscles in the following experiment, in which the muscles became part of the reinforced pattern. So in this situation, there were 4 groups of forearm muscles that were recorded: biceps, triceps, wrist flexor, wrist extensor. And the activity was rectified, and for each muscle it was multiplied by a certain gain factor, and then a unit in the cortex was also part of this. And the summed activity was then fed to the integrator, which had the same relationship to the meter and the feeder and everything. So now, in order to drive the meter to the reinforcement level, he had to generate the right pattern. And of course, what we were doing was tweaking these polarities and the gains to extract particular relative degrees of activation of the cell. And so here's an example in which this paradigm was used to reward the monkey for contracting in isolation, relative isolation, each of these muscles. So actually, you can think of these as muscle groups. So for example, for the biceps, the plus here means the bicep activity drove it to reinforcement. The minus for flexor, extensor, and triceps means that any activation would have prevented the reward. And the unit activity had a 0, meaning it wasn't part of this contingency. So here the pattern is activate biceps, suppress the others. And so he activated the biceps, and the unit went off with it. And then this is the same thing for a wrist flexor. So you can see that the wrist flexor in relative isolation also activated the unit to some extent, not as strongly, but to some extent. And then for wrist extensor, there was no unit activity. For elbow extensor also. So that was the basic idea. So there's a whole set of cells that were recorded with this paradigm. And one of the interesting things is that here the correlation between the cell and the muscle could be demonstrated a number of ways. First of all, it could be demonstrated by rewarding the monkey for contracting the biceps. When the monkey flexed the elbow, the biceps was activated along with the cell, as well as these muscles. And when the monkey was rewarded for isometric unit bursts, that is to say, the unit activity was rewarded, no contingencies on the muscle, he again co-contracted the muscles and with the unit, primarily the biceps. So it's a relatively consistent relationship. So this is obviously an invitation to test this relationship. So in this next part of the session, we reinforced the unit activity without the muscle activity. And lo and behold, within a few minutes, same amount of time, it took the monkey no time at all to just activate the cell without the muscles. So at the time, this was actually a little bit surprising because everybody was thinking that these motor cortex cells had a very strong obligatory relation with muscle activity, but this says you can dissociate it. And the monkey can dissociate as long as it pays off, as long as you've rewarded the monkey for doing this. So another paradigm was the opposite. Can you activate the muscle without the cell? We didn't actually get that far in this experiment because the monkey, well, he started to work in that direction, but he was getting less and less interested in applesauce by that time. So it may be a behavioral thing. Now, today, we've reinvestigated this dissociation of cells and muscles on the level of the CM cells, which have direct correlational linkage to muscle. And you can dissociate the activity of a CM cell and its target muscle in both directions. The monkey can activate the cell without the muscle and vice versa. So in retrospect, this is actually easily interpreted in the context of the fact that these cells in motor cortex are often involved in mental imagery of movement. You don't necessarily have to make a movement, but if you think about it, you're using many of the same cells. And this is possibly what this animal was doing, is just thinking about it, but not performing it. But the interesting thing is how rapidly you can dissociate thinking about a movement and actually performing it, which is a good thing. We don't act out everything we think about.
But do you see this as a specialization of M1, or do you see it really more as a generic property of cortical neurons?
Which, the dissociation?
The dissociation, yes.
I think it's probably generic. I think everything is very flexible. In this case, we're talking about a system, the motor cortex, that has a relatively direct, and in this case, consistent relation with the muscle. But if you probe it, then you can get this dissociation. So I don't know what the analog would be, like in sensory cortex, maybe.
You can remap a receptive field, right?
Yeah.
The theory. Yeah. That this binding is not fixed.
Yes, that's a good,
and that's
good
true.
experiment. That would be interesting to do. Yeah.
Well, what you know in the sensory cortices is that you have your thalamic projections that put some constraints on that.
Right.
But within those constraints, you can sort of freely move around.
Right. Turns out that we did this in sensory cortex, postcentral sensory cortex, which you would a priori imagine the monkey would drive by stimulating receptive fields. Turns out the peripheral input is only one of the inputs. That's the classically identified one. The other input is this volitional input, because these cells fire before active movement, and if you reward them in this way, monkey will activate the cells without any movement in postcentral cortex. So it depends on how you probe the system, how you ask the question. Yeah.
It's very interesting. You could imagine that the basal ganglia is maybe involved in gating, so that when you want to imagine the movement but not make it, then the basal ganglia is suppressing the motor system. But what I find surprising is that you could move one muscle but only imagine moving another muscle, because that intuitively sounds difficult.
Yeah, so that's a good idea. I think, I'm trying to remember, I think people are now beginning to probe. Actually, Chet Moritz in our lab is asking a monkey to make one movement and then volitionally controlling cells to move the cursor with another simultaneously. Yeah. So that's a work in progress.
I understand why you would use monkeys for this, but I wonder if you tried similar level of control and discrimination, say on a rat. Would you be able to train them to the same extent? So,
definitely. Yep.
Yeah, how fundamental a property of neurons?
It's probably any animal that moves around can probably, and has a reinforcement system, can probably be a successful participant in these sort of experiments. In fact, Jose Carmina did a wonderful experiment showing that rats with auditory feedback can learn to volitionally control patterns of activity in motor cortex. Actually the very first experiments were Jim Olds did this in midbrain. So, is this? Yeah, it's actually here. So these sort of experiments are part of a whole raft of biofeedback conditioning of central nervous system activity that was done in the 70s, I guess. And this involved conditioning activity of single neurons, motor units in the arm, isolated motor units, motor cortex cells. We did that, and Ed Schmidt did that, and midbrain rat. This is the Olds experiments that really pioneered this. One can also volitionally or do biofeedback studies on control of EEG and local field potentials. Cortical alpha was all the rage because people thought this was some correlate of some mystical state. Turned out to be just a correlate of rest. Hippocampal theta studies were done. Amygdala spindling in a chimpanzee was a wonderful experiment by Jose Delgado. And what he did was, this is an experiment you couldn't do today. Number one, because it was a chimpanzee and it was invasive. And number two, because the stimulation was aversive. But nevertheless, he showed that if you implement a loop from amygdala spindling to this reinforcement site, negative reinforcement site, the monkey, not the monkey, the chimpanzee controlled the amygdala spindling. And then one can also see that evoked potentials could be volitionally modulated up and down in visual cortex, auditory cortex, and more recently, Wolpaw's done some nice work with spinal stretch reflexes. So these are all experiments that predate the brain-computer interface, but they've all disappeared over the electronic event horizon. So they've now been reincarnated in these brain-computer interface paradigms, which is essentially the same thing, except now the feedback is provided through these external devices. And as I think everybody in this room knows, you can get these brain signals from various levels, from the surface of the scalp in the EEG, from the surface of the brain in electrocorticogram, and then from single units or local field potentials recorded intracortically. So you have this spectrum of greater specificity and resolution of the signals that's associated with also greater invasiveness of the recording technology. So any of these signals then can be processed, transformed into signals that control the position of a cursor on a screen or the position of a robotic arm, and it's the job of the subject to use the feedback to volitionally optimize this control. So that's your basic brain-computer interface. You can also use these extracted signals to control a stimulator that can deliver electrical stimuli into the muscles or into the spinal cord or back into the brain at various sites. And so this is what the recurrent paradigm that we're doing. So this bidirectional brain-computer interface is this part of the loop, and all this stuff can now be put on a little circuit board and operate continuously during free behavior. And what we've called this a neurochip. So what it consists of is a printed circuit board that's populated by off-the-shelf components, amplifiers, little computer. It's connected to electrodes, typically in our case in motor cortex. And so the amplifier feeds a little computer chip, printed system on a chip from Cypress Microsystems. That has an A to D converter in it and can be programmed to do a number of things. And one of the things it can be programmed to do is to detect when the recorded signal exceeds a threshold and goes through two dual time amplitude windows, and that's identified as an action potential by this template, and that acceptance pulse can then be stored to memory or used in real time to trigger stimuli that go back into the brain at another site. So in the recurrent mode, this loop is operating continuously, triggering from spikes, delivering stimuli. And there's recordings are made from a set of wire electrodes that are really remarkably stable. They're in the motor cortex, And here's an example of recordings of the same action potentials of the same cell for 14 days during day and night cycles. There's a whole interesting business about how these cells and muscles are related during wake and during sleep. That's in this paper. But these recordings are quite stable. Unfortunately, I'm prevented from showing you a wonderful video with a monkey jumping around, and you can see these action potentials being quite stable. But take my word for it that this is really possible during free behavior. With the battery-powered, this electronic, I should have said, is battery-powered. The monkey can carry this around on his head, and with each iteration, this neurochip is getting bigger and the monkey's implant is getting bigger, but nevertheless, it's still possible. So it operates, as I say, during day and night free behavior. So what is all this good for? So this recurrent brain-computer interface can be seen to implement two types of applications. One is to create this artificial recurrent connection. And as we saw, the brain adapts to consistent sensory motor conditions and could conceivably learn to incorporate this recurrent brain-computer interface into normal behavior if it makes sense. And that has prosthetic implications because the artificial circuit could be used to bridge a lost connection in the, with spinal cord injury, for example. The second type of application is to create synaptic or Hebbian plasticity because spike-triggered stimulation strengthens synaptic connections. And this again has clinical applications in strengthening connections that are weakened by stroke. But we're just talking here now about the basic science aspects of it. And I'm going to show you one example of this and three examples of this. So an example of creating this artificial recurrent connection was explored with Chet Moritz and Steve Perlmutter in an experiment that allowed the monkey to control functional electrical stimulation of muscles to get a cursor into a target. And the way the experiment proceeded, first the monkey was fine, a normal control of muscles, and he got the cursor into the target by contracting the muscles and generating torques that controlled the cursor, and he got it into the target. Then there was a nerve block applied that paralyzed the muscles, so the monkey could no longer do it through volitional signals to the muscles. And instead now the cursor position was controlled by the firing rate of the cell. So we quickly transitioned to the possibility of, to the fact that you could drive this cursor into the target with cell activity. Then the cell activity was connected to a stimulator, and the evoked twitches were used to generate torques that drove the cursor into the target. So here's an example of a session in which a monkey acquired different force targets. These are extension and flexion targets. These are the forces generated through functional electrical stimulation of muscles. These are the firing rates of cells. So these were two cells. One is connected to stimulating flexors. The second is connected to stimulating extensors. And the stimulation is determined by, first of all, crossing a threshold, and the intensity of the stimulation after that was proportional to the difference between the firing rate and the threshold level in order to get graded force. It's much more efficient than just doing rate, changing the rate. So the monkey, when he saw this flexor target, activated this cell, passed this threshold, and got his reward. And when he saw the extensor target, he activated this cell and stimulated extensor muscles to generate these torques. So this is sort of like that operant conditioning paradigm, except now it is mediated through this nonlinear transform from cell activity to forces.
Would you say that you could do this with any arbitrary set of muscles?
Arbitrary set of muscles.
No,
Yeah, I would say so,
case,
but this is a setup because these muscles here are muscles that he's used to.
Right, exactly.
Right. Yeah. I think you could, but you'd have to basically have more time to learn and set it up in a way that reinforces the particular muscles that you're interested in demonstrating this for.
Okay, so you could do this with abdominal muscles or...
Yes. So there's an interesting paradigm there. You're thinking about a breathing system?
For instance, but the key thing is here you have a bias that you already are controlling muscles that have to do with the manipulation of objects in the world, right? So this is already a channel of action. But if you can show that you can do the muscles that are completely arbitrarily related to the task, then of course your bandwidth would jump up.
Right. Yeah. Good idea. Yeah, I'm guessing, it's obviously just a guess, but I'm guessing that if the muscle can be somehow modulated in activity, this will work.
It should do it.
It should work.
Yeah. Exactly.
Yeah, just supporting that line of thought. Right? You could think in the case of diseases, there's a practical implication like motor neuron disease or multiple sclerosis, where function is being lost progressively and it varies between individuals, so you never know precisely what muscles remain in good function in a subject. So you could use your toe, or you could use your abdomen, or you could use whatever to do sophisticated tasks.
Yeah, I think so. In principle, that's possible, I would say. All right, so let's see. This is bidirectional movement with two cells. You can also do it just with one cell by having flexors stimulated when activity of that one cell exceeds the positive threshold, and stimulating extensors when that cell's activity goes below another threshold. So now the monkey is increasing and decreasing cell activity in order to stimulate flexor and extensor muscles. So one of the interesting things is that, getting back to these questions that were just raised, is that it doesn't really matter whether the cells are related to movements of the wrist or not. So we looked at that in the terms of the directional tuning of the cell. So here we have a 2D plane with flexion, extension, radial, ulnar, isometric torques. And a tuned cell would be one that has more activity in one direction, movements into one targets than into others. And about a third of these showed some tuning, and 2/3 of them weren't really tuned at all. And in the initial phase where the monkey was allowed to acquire the targets by cell activity, there was a slight advantage for the tuned cells compared to the untuned, probably because the targets were actually presented in the preferred direction. But once he had learned to control the activity of these cells, it didn't make much difference in terms of how fast he was acquiring the targets. So the take-home lesson is that this biofeedback stage before connecting to the muscles actually increases the number of cells that you could use.
What do you know about the cells that you're using here? Are they, what layer are they in? Are they corticospinal neurons?
Yeah, so all we know is that they have nice big action potentials that can last for the session. So they're probably layer 5 cells.
Right.
But we don't have any direct information about their projection. I'm guessing a lot of them would project to the spinal cord.
But Eberhard, there's of course a caveat in that conclusion, right? Because this might be true given the fairly restricted number of degrees of freedom you're controlling. Imagine you would have to control a larger number of degrees of freedom of the system, then maybe you need a certain level of, let's say, redundancy or a certain coding size to do this reliably.
Definitely.
Right?
So there's limitations. I'm going to tell you even more problems with this. This is just the proof of concept, actually, that you can take cells and trigger functional electrical stimulation of muscles, and maybe one of the simplest ways is just to connect them directly, as opposed to a more complicated way, which is to decode, first of all, go searching for cells that are related to the movement you're trying to create, which is totally unnecessary, I think, because you can learn to volitionally control these cells in new ways. And then the decoding agenda also then has to re-encode into muscles. So I think given the possibility of having long period of time to learn this direct relationship between a cell and a muscle, this could work. You might worry that this is a huge, what do you call it, cognitive load, but it's not a problem. Well, I showed you those experiments that show it's probably not a problem. But there are problems with this, and that has to do with what Paul just mentioned, is the limit in the degree of control of lots of muscles, particularly the distal ones, and how do you control patterns of activity in these cells. It's very awkward. But the second problem is that electrical stimulation in the periphery recruits the muscles, the motor units within the muscle, in the wrong order. It actually recruits the large motor units first. They fatigue readily, and so getting graded force is a challenge, and keeping these electrodes in for long periods of time is a challenge. So these are issues that made us go to another area where some of these problems are solved, which is in the spinal cord. So if you stimulate in the spinal cord, you recruit fibers that recruit the motor neurons in the normal sequence from small to large. And also, you actually typically at any one spinal site recruit muscles in some sort of synergistic fashion. So a very common response to stimulating in the spinal cord is co-activation of the thumb and the finger, for example. So we've done this experiment, haven't analyzed the data in sufficient detail to say anything much about it yet, but it does work, and the monkey can learn to use this loop. So the best thing we've done along this line, the closest thing I should say, not the best, but the closest, is to demonstrate that a monkey can use cortical activity to gate spinal stimulation. So this just came out in Frontiers, Yukio Nishimura and Steve Promoter again. And what happened was that a monkey had an unfortunate accident in surgery which made him paretic, so he couldn't really control the hand anymore. He had learned this task before the surgery, but afterwards he couldn't create the forces to get the cursor into the target. So what Yukio did is have a loop from the power in the high gamma range. So this is sort of filtered high gamma activity that can exceed a certain threshold. When it does, it triggers electrical stimulation of the stimuli aren't shown, but these vertical bands show when the stimulation was applied, and that generated torques, and the monkey got the cursor into the target by just simply increasing the gamma power. And then there were catch trials where the loop was opened and he couldn't acquire it, even though he tried.
But wait, to understand it, so you have to move the cursor, right? So I have to exert forces in at least two directions.
Actually, this is only one direction.
Ah, okay. So
Yeah,
it's,
yeah.
okay.
Yeah,
So it's
yeah.
gamma power.
This is a very simple experiment.
So do 1 degree of freedom here.
Yes,
Okay.
that's true. Right. So very simple. It just demonstrates that you can get control intraspinal stimulation by power in the high gamma.
How rapidly can the monkey really modulate this voluntarily, the gamma?
Pretty rapidly. I think that you have to remember the caveat that the monkey had already learned this task before he was hooked up to this. So one question is how long does it take him to learn otherwise? But
if it's
we're not in the
performing
business. I beg your
So
pardon?
if it gets the go signal, it sort of immediately starts.
No, this is all on his own. It's self-paced, pretty much. Yeah. So, this is, like I say, a very simple experiment. I just threw it in because I think it speaks to the point of getting cortical signals to control spinal stimulation. But the single unit data is coming up as soon as we finish analyzing.
You think that in a human patient, for example, the equivalent of having learned it before is to understand the task. Suppose that you tell a patient, you've got to move that cursor,
Yes.
and then person tries to
Right.
connect. Would it work the same? Is that?
I think it could work the same. And you just have to have a signal that the human subject can actually control. And then they would learn to control it. You might give hints if you know that the place where you're recording the activity is related to imagery of a certain type. You know, that's typically what happens, and then...
Because the other connecting kind of stream of consciousness that comes to me is whether the monkey can generalize the same control of the cursor for some other task. Suppose that the monkey now wants to move the cursor in a circle or move it up instead of to the right. And can it use its acquired skill for controlling it in one task to do something else?
That's a good question. I think I'm trying to remember experiments that speak to this, but I think Andy Schwartz has very sophisticated robotic hand movements that can be controlled by brain activity in monkeys, that in humans too, that demonstrate control of a lot of degrees of freedom. So it's possible. One of the important conditions is the ability to learn that control over a sufficiently long period of time. It may not be obvious in the first hour or two, but if you had an implant that operates for days with the same signal, it's remarkable what you can learn.
So, in the case of spinal injury, you're trying to connect motor cortex up to the lower part of the spine. I mean, what about just bridging the bit of the spine that has the damage? Is that an option?
That is an option, yeah. So we would predict that you could actually bridge this through a direct wire from, let's say, a cell to a spinal site.
Yeah, but you would still go from cortex. You wouldn't.
We would go from
spinal.
cortex.
You wouldn't do. Tony's asking, I think, for spinal, right?
Yeah.
Oh, spinal. No, but you could go, and this paper also shows the same sort of paradigm with the signal acquired from muscle. So in a sense that speaks to your question, because the muscle is controlled by motor neurons in the spinal cord, and that feeds back into the spinal cord. So in that way, that could work. Well, recording in the spinal cord is pretty tricky, actually, because of the movement issues.
No, but also the thing is, here you have your volitional signal embedded in gamma, something you can measure, and it's not so obvious what you would have to measure at the spinal cord level.
Yes,
to have volitional control.
Right. So ideally, what you want to record is units, but units are not very easily recorded stably especially during movement conditions. But
presumably, if you do it the Tony way, let's call it, you would need spontaneously triggering neurons in the upper section of the reserve section of the spinal cord to be sent up to the cortex for future control, so that they can do the right thing. And so what you have in the cortex is that each of these neurons that you pick are heavily controlled by lots of other things, and volition can be applied to them. Now, that would be much more kind of polysynaptic circuit, wouldn't it? Because it's not really jumping from the upper section.
Well, that's a good point. I think you have other circuits to consider in this paradigm than the direct one. You have the descending control of the spinal cells that you're recording from, as well as the ascending links from that cell. So it's complicated, but the nice thing is the brain can deal with these complications without worrying about all the things we worry about as to how they do it. You just have that goal, you want to achieve it, and you do what it takes, and probably the right guess is that it'll eventually work, given enough time.
But one question, how dependent is this on the stimulation site in the spinal cord?
Very much so, because the stimulation site has to evoke activity in the muscles that are relevant to the task. And so that's a given. I mean, that's a...
But you really have to find exactly the right motor neurons that do that.
Right.
Okay.
So it turns out, when we did the mapping of the spinal cord, it turns out that there's no obvious simple map in the spinal cord like there is in the cortex. And you have to go hunting for the site that activates muscles that you're interested in. And that's because what you're stimulating are fibers, and the spinal cord has a complex mixture of fibers. And
Right, exactly.
yeah. Okay, so then moving on, we have the possibility of having recurrent loops from one brain site to another, conceptually, and what this actually turns out, we tested this in motor cortex, and this is the segue to the first plasticity example. So in this case, wire electrode, this is actually a picture of wire electrode. This is study Andy Jackson, who's now back in England, and Jaideep Mavori, who is an electrical engineering student, combined their talents to make this experiment work. So the microwires are shown here. Basically, they are 50 micron wires that are advanced by this microdrive. Your finger, little forceps, can actually manually push the wire down. When it gets to a cell, you can stop. And then if next day you might have units, and when you have units, they usually stay around for quite a while. So the experiment involved recording from one wire and stimulating through a neighboring wire at another site. And so the recordings of action potentials were obtained at a site called NREC for recording, and that triggered stimuli delivered at a stimulation site. And this loop was closed for 24 hours or more of supposed to represent free activity of the monkey, with all of this operating up here in that chamber. And so it turned out that this produced changes in the motor cortex. And the changes were identified in terms of the output effects obtained from these three sites. Well, these two sites, but then there's a control site which was just used to see whether you needed this connection or not. So the outputs were determined with the monkey seated in a chair at rest, and forces were transduced that were evoked by about 300 pulses per second. I forget, 11 pulses, something like that. Standard train of microstimulation evokes movements and muscle activity. So, for example, stimulating this NREC site activated the FCR muscle and a little bit the FCU muscle, and also in the isometric flexion extension radial ulnar plane, you got trajectories, little torque twitches that went in this direction. And then the other sites similarly, NSTIM activated ECR, and control site activated FCU, and for each of these sites the isometric torque trajectories are shown here. So the dotted line is the actual trajectory, the arrow shows the average of that trajectory. So these were three sites that had different outputs in this train of microstimuli delivered with the monkey at rest. So this is the condition before, preconditioning outputs from these sites. And then the loop is closed. This is actually a picture of the action potentials recorded, and the stimuli were delivered with a delay of about 5 milliseconds. You see a stimulus artifact here, but pretty soon you can record again these action potentials. So the stimulation didn't actually produce any muscle responses, or didn't disrupt the normal behavior of the monkey. You could see when the cell activity was at a certain rate. These intensities, by the way, were on the order of tens of microamps. So when the cell was bursting, you could see little twitches sometimes, but otherwise it doesn't bother the monkey. So this is running continuously day and night, and then the outputs are tested again, the same way with the monkey at rest, and most everything is the same. The control output is the same muscle. The stimulation site has the same output, but stimulating this rec site now activates this muscle, which is the same as activated from this site. And what that did was change the torque trajectory to something like this. So these two torque trajectories and stim control are the same, but the NREC now generates a movement that goes around like this, and this is the average of that trajectory. So basically that's because the stimulation here now activated the muscle that was previously just activated from this side. Now, one of the interesting things about this conditioning effect is how long it lasted. So if you plot these directions of torques over days, before, during, and after conditioning, you see this, that the directions for the control and the stim site don't change, but the direction evoked from the recorded site changed in the direction of the end stim site, like this. So these were 2 days of conditioning, and then it lasted, surprisingly enough, for about 8 days. It's relatively unusual for things like this to last this long. But nevertheless, it lasted,
But how do you explain this effect exactly?
Okay, here we go.
Good.
Here's your answer. So one way to explain it is that you have these three outputs before conditioning. You do the spike-triggered stimulation, which induces synchrony between the recording and stimulation site, and that could strengthen this synaptic connection. So that would explain why afterwards, when you stimulate here, you get this output in this muscle.
You have a control condition where you had them out of sync and there was no learning.
That's right. So that's a good point. So the control outputs didn't change, which is a control for the effect of stimulation itself. Mm-hmm. It's not just excitability changes that are general, but it's actually this connection. So it's even better than that. Actually, it depends on the time between the spike and the stimulus. So in a bunch of experiments repeating this with different delays, the change in the mean torque from this site was obtained only for a spike stimulus delays of 75 milliseconds or less. So this is a window of maybe 50 milliseconds where this conditioning paradigm works. And that's of interest because it actually is consistent with the window of spike timing dependent plasticity, which says that if you activate a presynaptic input and then a postsynaptic input, you strengthen that connection if they're activated within 50 milliseconds of each other. Is that?
What is the latency? What should be the latency through the...
So the latency, you mean the spike stimulus interval?
Yeah, I mean, when the presynaptic spike would get to the... So
well,
when the
we don't know.
The spike on the presynaptic neuron and reaching the synapse of the...
Well, we don't know in this case. It would be great to be able to actually record that spike-triggered response, let's say in the post and the other cell. But they're too close together. The latency is so small that the stimulus artifact obscures the evoked response. So we don't have that information in this study, but we do have it for the third study. I'll show you.
So, but again, it would be very small, like less than 10 milliseconds, or 2 milliseconds.
Yes, typically, right. Yeah. So, right. Mm-hmm. All right, so let's see what else. Oh yeah, okay, so this is the summary of a whole bunch of these experiments showing these trajectories before and after conditioning, and in every case that's shown here at least, you see a change in the output from the NREC after the conditioning. So this is the most extreme example where NREC output was in this direction before, pretty much opposite the end stim, and after the conditioning it flipped around and the output went in the end stim direction.
But now end stim also changed in its response.
The end stim also changed its response, yeah. So it's more complicated when you look at the details. So here the end stim was reduced. That's what you're getting at?
Yeah.
But, little hand waving says it's going in the right, same general direction. It certainly didn't change as much as this NREC output. So if you plot the difference between the NREC and the NSTIM vectors before and after conditioning, the NSTIM minus NREC vectors, then this particular session is down here. It's probably the best one, where the difference is very big before conditioning and very small after conditioning. And this summarizes all the rest of the experiments that were done with the right latencies. And you can see that it tends to drive this difference in the vector directions down, but not always. I mean, some of these are not significant.
Distribution, right?
I beg your pardon?
It looks bimodal, as if you have at least two different classes of cells. There's some that are sort of hyperplastic, like the top example there, and another group that's really much more resistant to change.
Yes.
Would you interpret it in those terms, or you see this really as one population, one fairly homogeneous population of cells?
Well, yeah, that's a good question of why it didn't work for some pairs. Yeah. And we didn't see any systematic explanation for why it didn't work in some cases and it worked for others. Mm-hmm. They had very similar sorts of parameters. Okay. But you can see that it didn't work at all for any of the control sites. So the difference between the control direction and the NSTIM direction was pretty much along this line, before and after being the same. So I think it'd be interesting to explore this a lot more in terms of answering your question, also possibly in terms of making this a more robust conditioning effect. Okay, so that's example one of conditioning, plasticity. The second example is very similar, actually. So in this case, Tim Lucas did the experiment of testing whether instead of recording from the cortical site, you could record from the muscle that the cortical site is associated with. Otherwise, it's the same sort of paradigm. You have the recording site, stimulation site, and then the control site, each related to different muscles. And in this case, the muscle activity was used to trigger stimulation of NSTIM, and then the outputs were tested before and after that conditioning. So the rationale for this is that the muscles are a much more robust and less invasive source of signals, or get the signals less invasively. There's no guarantee a priori that this could work because of the timing, this is, but it actually did work. So here's the experiment, one of the results showing that the effect that you evoke from the NREC cortical site goes into the direction of the NSTIM site. And then at the end of, so this is all during conditioning, but then at the end of conditioning it doesn't last as long. So it's a good question as to why that doesn't last as long. But so don't ask me, I don't know. Actually, I should ask if anybody else has a good idea. I think it could have to do with...
as opposed to the long-lasting change you see at the cortical level, you mean?
Yes, exactly. That's right. The cortical duration is sort of anomalous in some ways.
Well, you could argue that this, of course, sits at the output of a much larger cascade of neural responses, which have themselves tuned to specific actions, muscle patterns, and so on. So what you see here is not a direct result of the stimulation at the cortical level, but how this percolates through that whole cascade, translating to your muscle response.
Right.
That's what I would say.
Yeah, and there could be a lot of variations between the cortical site that's being tested and the muscle. So
exactly right.
Yeah.
Then there's other systems that say, hey, wait 1 moment, we don't want to move there, we want to move there.
Right.
Right? This is roughly what's happening then. You could argue.
Yeah. Yeah, I think something like that. So here again is a summary of these experiments showing that the angle between the NSTIM and the NREC output decreased after this sort of conditioning relative to before, and not so much with the control sites, except for, in this case, there were some control sites that also changed. So that's, oh, this is wrong. This is actually out now in Journal of Neuroscience. I should update my slides here. So do I have it here? Yeah, there it is. So this whole thing is in this paper.
But you're only talking about the direction of torque, right? But do you also measure force?
Yeah. Well, this is force. It's isometric force.
Oh, okay.
We're talking about. Yeah. It's isometric force in a vector in this plane.
All right.
Actually, in this study, the planes were a little bit more long story, but anyway, it's force output. I refer you to all the gory details in this.
All right.
Yes.
Just one thing. The first drop that you have in the stimulation and then the raise is
This drop?
times in the stim, the blue line.
The blue line, yeah. So there's a lot of variance here. That's true. I can't explain that.
Okay.
I think there was variance that is part of the experimental condition. I have no interpretation or explanation.
Sorry, one question.
Yes.
Well, I don't completely understand the paradigm, but would it be possible to do reacquisition in this?
Yes, yes. Actually, there's some interesting data in this paper about the fact that you can not only reacquire, but you can actually maintain the output effects with less and less stimulation once it's done.
One explanation for this drop would be that you have this learning, but then you have an adaptation by another system
Right.
that goes in the direction. And then next time you have to, when you readapt, when you reacquire, you have to move away the adaptation, but not learn the.
Mm-hmm. Yeah, there's some interesting questions as to why plasticity reverts. Well, let me get back to that issue, yeah. All right, so that's example 2. Here's plasticity example 3, which has to do with the strength of corticospinal connections. So in the non-human primate, in the human primate as well, there are cells in the motor cortex that have relatively direct connection to motor neurons that control muscles. Talked about this yesterday afternoon. And these cells, when you do spike-triggered averages of rectified EMG activity, they demonstrate a post-spike facilitation of this average EMG activity. This is about a 30 millisecond sweep. This typically peaks about 10 milliseconds later, consistent with conduction to the spinal cord and probably a relatively direct corticomotor neuronal connection, which we know exists from intracellular studies of the EPSPs produced in these motor neurons from cortical stimulation. And this can appear in multiple muscles, so these cells have divergent connections to multiple muscles. And, well, that's shown here actually. This is a summary of the sort of connections that are obtained in these studies in terms of the muscles that are facilitated from a cell, and they're also inhibitory post-spike effects and antagonists of these facilitated muscles. So you have basically this sort of circuit going on with post-spike facilitation produced by excitatory connections to motor neurons of the excited target muscles and inhibitory linkage to antagonists of these. So the idea is to see whether this spike-triggered stimulation could be used to modify these connections. So the experiment went as follows, as Yukio Nishimura and Steve Perlmutter performed 2 miracles. Number one is to actually put these wire electrodes right by one of these cells and record the activity of the cell for days, same cell. And the second is to use those spikes and use the neurochip to deliver stimulation at spinal sites that activated the target muscle. And the timing between the arrival of the descending activity and the neurochip stimulation of the postsynaptic cells, that is to say the motor neurons, could be controlled by the neurochip. So this paradigm actually worked. So these so-called post-spike facilitation effects were measured in a standard situation. The monkeys doing a target tracking task a certain level of EMG that allows us to get the spike-triggered stimulation of the EMG, measure this post-spike facilitation. This one shows, okay, and then this overnight conditioning in the home cage was done for, in this case, almost 22 hours of spike-triggered spinal stimulation. Again, there was no obvious movements that were, the intensity was not that high that it actually perturbed the movements in any obvious way. But then testing again the second day under the same conditions, has to be done under the same level of EMG activity. Then the spike-triggered average produced a larger post-spike facilitation. You can measure this as the so-called mean percent increase of this feature above baseline. So it was 6 before and it was 10 afterwards. So this increase actually represents an increase in the strength of the connection from this single cell to the target muscles, target motor units.
But wait, at this stage you could argue it's a non-specific effect, because maybe if I'm boosting the whole circuit, that's possible. So how can you...
No, actually that is controlled by the fact that these were compiled under the same behavioral conditions. Oh, okay, no, I know, stimulation could act, yeah.
So you facilitate the whole circuit.
Great question. Okay, I'll be back with the answer to that.
Okay, good.
Okay, so these are showing these post-spike effects, again, under the standard behavioral condition of target tracking. First, without any conditioning to show how that these are relatively stable. And then after about, in this case, again, 22 hours of spike-triggered stimulation, you can see that the post-spike effect goes up. These are the mean percent increases. And in this case, the cell facilitated two muscles, and they both went up. It inhibited an antagonist, and that also, the post-spike suppression also increased. And then another day of conditioning, this went up a little bit more, this went up a little bit more, this one disappeared. Okay, so that's experimental reality. And then after a day of no more conditioning, in this case, this feature stayed above preconditioning levels. This one didn't, these reverted. So there's an interesting question here. In some cases, the features lasted for 2 days. In other words, there was another day of non-conditioning, but testing again, and these changes lasted. But you can see that there's a trend for these effects to revert. So this is an interesting question of why would it revert, actually? I mean, what's determining the steady state values of these post-spike effects? There must be some mechanisms that determine if it's too large, it has to go back. So I don't know what that answer to that is. But
also the gain of the increase is not exactly matched, right? So
that's right.
You see much larger increases in the neural response as compared to the response in the muscle. So the muscle...
How do you mean, the neural response?
Well, if you look at these changes here, right, in the neural response or
which
the muscle response.
Which one do you...
Well, you have there day 1, 11.68, coming from 6.77, right?
Right.
So that's almost doubling. But on your next row, which is more related to the muscle, if I understand it, this increases maybe, let's say, 0.6, 0.5.
No, these are 2 muscles. These are 3 muscles. Actually.
Oh, they're all muscles. Okay.
So these are, this is one muscle on these successive days, post-spike effects. This is the second muscle on the successive days, post-spike effects. And this is the antagonist showing suppression on these successive days. So,
but then the gain also in these muscles is not similar, even though you are... You would supposedly boost a single circuit.
Yeah. So that's you're thinking way too quantitative for this particular technique to answer detailed questions like that. Just to see the effect, I think, is the basic thing. But if
but it raises questions about how these circuits are organized, right? This is what I'm after.
Right.
I'm just trying to think of your, in general terms, your question about why going back, why would the effect be lost?
Yeah.
And one thing that comes to mind for the overall brain rather than for muscles is this idea, I think it's from Linnaeus, wasn't it? That he was kind of trying to view the brain as something which is continuously active with things that you could call noise from the point of view of action and firing and everything else. And everything you learn is kind of taking you away from this basic state, but dynamically, so that if it doesn't work anymore, then you go back to talking to yourself.
Yeah.
In a way, you need this spontaneous activity to actually train it. So you need the cells which are doing something. And if every time they get moved from that, they stay there, then you will be losing plasticity.
Yes, okay, so that's an argument.
The muscles.
Right, so what is it then that makes these strength of these connections to go back? Somehow they have to know how much they're being used and what the appropriate steady state value is. So in this paper, in the discussion, the best I could offer is you have this spike timing dependent, I haven't shown it yet, but this obeys the spike timing dependent plasticity rule. And let me get back to this. Yeah, I have to show you this. So anyway, this is the sort of experiment again summarized, that you have the preconditioning post-spike effects in excitatory and inhibitory linkages. The conditioning produces the stimulation of the postsynaptic cells, and the post-conditioning effect is an increase in post-spike facilitation and post-spike suppression, probably mediated by increases in these terminals. Could also be an increase in this terminal. So that's the experimental result in a nutshell. So the fact is that this effect also is dependent on the delay between the spike and the stimulus. So these are the changes in this mean percent increase that's obtained with this conditioning performed for different delays between the spike and the stimulus. You can see that there's an increase in the mean percent increase for intervals, again, here between 12 and something like 25 milliseconds in this window. But there were no changes for longer intervals. So this is the control that you were asking about for just the effect of spinal stimulation alone. Spinal stimulation alone doesn't work on this, producing this plasticity, unless it's delivered with exactly the right interval. So the stimulation isn't enough. And these are actually significantly not different from zero, and this is significantly different from zero. Asterisks aren't on this particular figure, but they are. So the other interesting thing is this zero delay result. This is actually significantly below zero. And so that is really cool because it actually is consistent with this bi-directional spike timing dependent plasticity rule. What I mean by that is if you stimulate in the spinal cord at zero delay, you're stimulating the postsynaptic motor neurons before the arrival of the corticospinal action potential. And when you stimulate the postsynaptic cell before the presynaptic input, that tends to reduce the strength of the synaptic connection by this sort of a rule. This is the weight change as a function of relative time. And so that's consistent with that, and it's significant. You can actually see it in the raw records. This is going to come out in Neuron, and you'll see all the details.
Do you get also this effect in the cortical paradigm when you are only in motor cortex?
No, we haven't yet, because the conduction time is, number one, too short, and number two, we can only, so then we can't stimulate before the arrival of the conducted action potential within motor cortex. But it's conceivable that if this paradigm worked for other cortical sites where the effects were long enough, or the latency between the effect was long enough, you could repeat this in cortex. It's a good question, but yeah.
I'm wondering about that you get the antagonist going in the opposite direction. That implies something about the spread of your stimulating effect, actually. What do you know about, these are microelectrodes that are stimulating in the spinal cord.
That's right.
So are you...
Or wire electrodes.
You're assuming that they are stimulating both more than one motor neuron, obviously.
Yes.
So they are reaching the agonist and they are reaching the antagonist.
Correct.
Postsynaptic.
Yes.
Okay.
That's right. So actually there's an interesting story as to why what you would expect from the inhibitory link. How am I doing for time, by the way?
You're doing great.
It's okay? All right. All right. So anyway, it's not a priori...
11 or 6, in case you...
No, we're fine, we're fine.
Oh, so actually we should be winding up.
We're fine.
All right. So the disynaptic inhibitory linkage shows the same effect. I don't have but there are curves like this for the inhibitory effect. And a priori you wouldn't necessarily anticipate that this rule is the same for inhibitory linkage. In fact, people have shown it's, for some inhibitory connections, it's the reverse of this. But anyway, empirically it turns out that it was, very similar to this for the inhibitory link. We didn't actually get data in this thing, but the data in here, in this region, was the same. Yeah. Okay, so actually should be winding down here. So there are applications for this new recurrent brain-computer interface that we talked about, mainly signals that were obtained from cortical neurons converted directly to stimulation of these sites. But obviously you can extend the list to include sources of signals from multi-unit activity, field potentials, EMG, actually I talked about EMG, electrocorticogram, and not just motor cortex, but any number of sites. And the transform could be direct conversion, it could be some computed function of this activity, could be a neural network, which is being investigated here, it could be modifiable. And the targets, it could include any number of things, brainstem as well, I didn't put it here, but a reward center. So this is an interesting possibility, is that you could actually stimulate a reward center contingent on this activity and have a portable operant conditioning situation. So to test that, we actually took EMG activity, converted it to stimulation of a site that was rewarding in the sense that the monkey would do a normal task to get stimulation there. So this was the thing. This was it. So it was nucleus accumbens at a site where the monkey would do alternate flexion extensions to stimulate the accumbens. And then Ryan Eaton and Stavros Zanos did some recordings from the EMG of the biceps. And the neurochip is programmed to go alternately 5 minutes on and 5 minutes off in order to get a comparison. These are the number of pulses that were generated to 5 minutes on and much less during the 5 minutes off. And in fact, watching the monkey, you could see that he was starting to flex the elbow during the 5 minutes on. Yeah?
Sorry, just curiosity, wouldn't it make sense also to do this from sensory areas, for remote sites, for instance?
From which side? Sensory?
Sensory areas from, let's say,
yes,
visual areas, just to see if this can
definitely. No, it would be very interesting. There's a whole new area. It's totally unexplored. Should do it.
No, so
but
my question is, would you guess it would have any effect? Or?
Well, I guess it would have an effect. And then the next question is, how does the monkey do it? So if it's a sensory site, for example, let's say extreme case of visual cortex, if you reinforce the visual cortex cell, you would probably see the monkey making eye movements that optimally activated the cell.
Yeah, but because you have you can maybe reinforce the color red, and then whatever task you do, the monkey could approach the red for some reason.
Yeah.
And then you give behavioral flexibility, you can approach with eyes, with the arm, or whatever. So,
Right.
do you guess it would be something similar to what I'm saying, or?
I think it could work. The next question would be how it works and how to analyze how the monkey's doing it. So once again, I think all we've explored outside of motor cortex is sensory cortex, so-called sensory cortex, post-central. But turns out those cells have this volitional input. So they're like motor cortex cells in that regard.
But
Okay,
you
so-
wait, Eberhard, did you map the EMG directly to the value? The intensity of the EMG was directly correlated to the intensity of the stimulation, or it was more like a switch? It didn't depend on the intensity of movement, how much
It
it
did
stimulated.
depend. I mean, these were pulses that were triggered from the EMG activity. So the more EMG activity generated, the more
But
stimulation
the
it
more
got.
events or the more amplitude?
Just the frequency.
Okay,
The amplitude was the same in this case.
okay.
So it was the frequency that was changed.
Would have preferred movement speed, or would just go wild and
Well, he did variable things. I mean, sometimes he would just sort of reach behind, and other times he was just... It was a very variable, but whenever he contracted his biceps, he would get this.
But they seemed like, let's say, goal-oriented movements.
Well...
Because you can if you're not a monkey...
No, not really. I mean, he was just learning that whenever he did something that activated the biceps, it felt good. So he did more of it. And he did this, by the way, let me just finish this. He did this, these are the 5 minutes on and 5 minutes off for almost 20 hours. So he continued.
It's pretty amazing.
Yeah, it is, except for time out here for sleep, probably.
So you're assuming that the accumbens is then driving the dopamine cells? Is that?
I'm not sure, I'm not an expert on that, but whatever it's, yeah, I think so, yeah.
Why didn't you just directly drive dopamine neurons?
Well, we tried the ventral tegmental area and had a lot of trouble with that. It didn't work easily. And so we just decided to go to test another site. And the accumbens was a larger target and it worked nicely and we stuck with it. But we have no real detailed information about how it's working, other than the fact that it's operationally rewarding.
Sorry.
So, yes.
In this particular study, I'm missing probably the main point. How does this differ from the very classical description of what self-stimulation centers are? I mean, you could have a rat.
Well, the difference basically is that this is an operant conditioning paradigm operating during free behavior. So the monkey is moving around in the cage. He's not in a task in which the movement is constrained. So why would that be of interest? And that's because under free conditions, where you have plenty of time to explore whether or not the monkey has volitional control over things, there would be a possibility of extracting correlates of neural activity that are occurring during free behavior that would take time to discover. What I'm getting at actually is the following. The coolest experiment I can imagine is testing this idea of temporal coding. So you know that the idea of temporal coding is that information is processed in the relative timing of spikes. And that's an idea that's in dire need of empirical support. But if temporal coding is actually being used, then if it's part of a volitional repertoire, the monkey should be able to generate it. So we should be able to reinforce a pattern and get the monkey to generate that pattern more frequently. If that can be done, that would be cool. Unfortunately, even a simple transition from the muscle to units has not been as robust as I would have expected. I have no idea why generating these stimuli from unit activity instead of muscle activity has been less robust. There's lots of complications. The reinforcing efficacy can decrease and so forth. So I haven't thrown in the towel, but...
Well, wait, but couldn't you argue, Eberhard, that isn't it obvious that it's more difficult to do it from unit activity? Because if you generate it from muscle activity, then you could argue that the pool of neurons recruited is much larger.
Yes.
So you have a much larger repertoire to play with.
Okay, yeah, but you could argue, as we talked about before, that even if you had it contingent on a unit, he's activating a lot of cells just as much for unit activity as he is for muscle activity.
You're just using muscle, right?
Yeah, but it's not a specific motor activity, even with unit being the thing that triggers the reinforcement, because it's activating lots of cells.
Okay.
So I don't know whether that's totally the explanation. But let me think. Yeah, okay. So this is this recurrent brain-computer interface paradigm is something that's being investigated here, as most of you probably know. And it's a wonderful experiment in which the computer part is actually something that does some interesting computing rather than just input-output. And this is, I don't know whether the students are studying this as part of the project or something, but anyway, it's described in this paper that you can hook a sort of neural network model that simulates cerebellar circuitry that mediates eye blink to the brainstem of rats that are anesthetized and demonstrate that you can get combinations of inputs from the pontine nuclei and inferior olive that are required to mediate the plasticity in the eye blink, and the output here is stimulating facial nucleus, if I understand this right, that produces the eye blink. And all of this learning is done on the chip that's in intimate interaction bidirectionally with the brainstem. Great idea. I'm probably describing it wrong, but that's the idea.
No, it's very good.
Okay. It was a wonderful experiment. So then one also has to acknowledge this relatively ambitious agenda of Ted Berger to also use VLSI circuitry to model neurons in a serial pathway. So he's interested in his first studies of replicating what CA3 does in this pathway from dentate to CA1. And so there's a VLSI circuit that has multi-unit inputs and triggers multi-electrode stimulation, and the idea being that you could potentially replicate the function of this element with a VLSI circuit that substitutes for a lesion here. So this would be like a cognitive prosthesis. And the idea is, I like this animation because it shows, again, how this could conceivably work. If there was a pathway from short-term memory to long-term memory via the hippocampus, and then there was a lesion in the hippocampus, then having a VLSI biomimetic model of this nice connection, recording the input and stimulating output, that you could actually restore that function. So this is very ambitious because I think it depends a lot on the ability to stimulate in ways that replicate what the neural circuitry does. But nevertheless, it's a challenging project.
But it's interesting also to see the memory model that Berger would have in mind here, right? It's really like a very clean, chunked pipeline.
Yes.
So if you go through a memory stack,
right,
it's in a computer, which is obviously not the case for these structures, right?
Yeah. So this, again, it's a serial model, or model of a serial pathway of transmission of information.
Yeah.
So I put it up also to point out that if you had a computational element here that's bidirectionally connected with the brain, the question is whether the brain could learn to exploit that computational resource. We're talking science fiction now, right? Okay. I don't want to squander my credibility here. But nevertheless, looking into the future, you could imagine that actually the challenge is to design this sort of interaction in a way that would demonstrate that the brain could do something interesting with this computational resource that is directly connected to the brain. So there is, I don't know, it's fun to think about it, and I'm guessing it's going to happen, and it's science fiction today, but as you know, what's science fiction today, this would have been science fiction a couple decades ago, this thing. But technology is increasing exponentially, understanding of the brain is increasing, blah, blah, blah. So Ray Kurzweil is one of the proponents of this fact that all of this technological advance is going to create unexpected new things. And I sort of think it's probably likely that what sounds like science fiction now is going to be implemented in the future.
But you know, Ray would be saying that Skynet will take over the world soon, right? And robots will just kill us off because we're obsolete. That's his trajectory.
Oh, is that where he's going? Well,
yeah,
maybe we don't want to go there then.
That's what I'm saying.
But
so maybe it's not just like, oh, something interesting will happen.
Yeah.
No, for him, Terminator will happen.
Okay.
And then he's saying like, and since this is very dangerous, you should give me all your money so I can protect you against Terminator. This is his program.
Really? So,
Yes.
so you've given him all your money, or?
No, but he is in the meantime also now appointed as the chief engineer of Google.
Yes.
So now you see where that's going.
Yeah. Ah, I see. Well, so there could be a whole lot of scenarios. Let's say that the world stays on track, and
Right, exactly.
and this technology was used to do this. It would be, I think, it's going to be very fascinating. A lot of the students here are going to see this happen, and many of them have the opportunity to contribute to make it happen. So, have fun. So with that, I think I just want to thank all the people who were involved in these experiments. Andy Jackson and Jaideep Mavuri had this cortical implant. Stavros and Ryan did this operant conditioning thing. Who else? Let's see. Oh yeah, Chet Moritz. And Steve Perlmutter was involved in a lot of these experiments. And hidden hero is Larry Shoup, the programmer who has to create new paradigms every time we think of something else we want to test. And he's a programmer. Tim Lucas and Yukio came after we were out on the beach. This is an outing on the Pacific coast. And I thank them and all the support from all these guys. And I thank you for your patience since I've gone way over time.
Great.
Thank you.
That's great. Questions? Tony?
So what's the state of the art and the likely future trajectory of really long-term chronic implants? So you mentioned 2 weeks is the period of good signals.
Yes, that's right. That's the period of signals for single units.
Right.
But I think COG, for example, and LFPs are recordable for much, much longer.
Do you mean years? I mean sort of decades?
Actually, I wouldn't, months, let's say.
Right.
We haven't really pushed it to years yet, but I'm guessing that you could get with LFP recording electrodes invasively, or better yet, surface cortical recordings less invasively. You would get reliable, repeatable signals for a long period of time.
Other questions?
Yeah, I got one question. You know, from the application point of view, there's some problem of enhanced manipulation here, because, you know, what if the object hesitates? I mean, thinks about doing something and maybe he doesn't want to do it. I mean,
yes.
You're describing the standard state of mind of our students. Hesitant.
Yeah. Yeah, I mean, like he would like to, for example, move an arm.
Yes.
And suddenly he hesitates. And, okay, I don't want to move an arm.
And
so he thinks about it.
Then the problem is
in enhanced manipulation, it moves the arm, but I don't want to do it. In fact, in a while. So
yeah.
the firing
so what's the
is
problem
before
here?
the movement. The firing of neurons is before the movement.
Mm-hmm.
So if I hesitate and I don't want to move my hand, the robot hand will move.
Right, okay, so the fact is that you'll learn to control the activity of your brain in a way that will smoothly operate this robotic arm, let's say. So you won't hesitate, or when you hesitate, it stops moving, and then you start up again. So I think I'm missing something here. Okay. But the sleep is the big problem. You don't want to have this thing flailing around with spindle bursts and stuff.
Ivan?
So, well, as you said, I mean, you are more doing these closed-loop chips where the chip is just transferring the information, and we have tried more to do the chip that does some learning.
Yes.
But I mean, in this talk, I was thinking that as you shown that the brain is so plastic and learns so rapidly, wouldn't it be possible to think of use the brain as the chip? I mean, you have a part that it's wrong, and then you, as in surgery, sometimes you replace one part of the body by another. I mean,
that's right.
So you could do use another part of the brain that's still intact.
Yes.
Learn the computation,
right,
and then use it as a bypass for a connection that it's not.
That's a good idea. That's a good paradigm.
We'll try it on your brain first, even. That's a great idea.
Well, what would be a specific experimental instance of that? You would be, for example, are you talking about like sensory substitution?
Like stroke, motor control. Right?
Yeah.
Oh yeah.
You have the motor control area that it's vision. Okay, so you
say what do you want to sacrifice?
Of the motor control, you take the inputs and the outputs of the motor control, you stimulate there, but you send them to some other area of the brain. I don't know. I mean, it's probably you have to think a little bit more.
You get a list. You can say, what do you want to lose? Vision, audition. You can.
I mean.
Well, yes, that's right. That's a great idea. And actually, the next generation of this neurochip is going to be much better than what I've shown you so far. It's going to have 16 channels of high resolution FPGA logic, many more loops and conditions that we can implement than we can actually think about.
Energetically autonomous?
It's also battery powered.
Okay.
Yeah, but it's getting, monkey's head is going to have to, the neck muscles are going to have to get a little stronger, but basically it'll work.
Cool. Stefan, you have a question?
So this is wonderful research that opened the way to a lot of new technologies, but why are you doing that on monkey? And how hard would it be to move to human? I mean, non-patient, but like people who would like to test things on their brain, enhance their brain.
Yes. Well, I think for humans,
are you volunteering?
Actually, I would like to go in this direction.
Yeah. So for humans, the population for which it's justified to actually put electrodes in is patients. And most of us would prefer to interact with these devices through normal channels. And that's really actually much more efficient because our brains are sort of used to processing a lot of information through vision and touch and so forth. So initially, but if you like to work your way in non-invasively, you can start with EEG. And there are now computer games that are EEG driven and so forth and so on.
But is there any real barrier from ethic committees, for example, that would prevent people to try that? Like people in your team who would like to implant themselves, could they?
No, they couldn't actually, unfortunately. I think the human protection agencies or whatever they are won't sign off on something that's experimental so easily. It has to have some clinical justification or promise, I think. I don't know. It depends on which country you're in, I guess.
So you're allowed to do it at home.
Right. There is an exception for the summer school, right?
Exactly. No, but the point is you always are stuck with these ethical protocols, and I'm sure you can volunteer as much as you want. These ethic boards will draw a line because they will say like, oh, the consequences will be unknown because Stefan is already impossible now, and God knows what like super Stefan, making friends all the time. So this will be no. I think it will be difficult, but at home, I'm sure you're allowed to do this. So we can give you some systems to take.
Yeah, I think it's a tradition in England and Australia. I think for neurophysiologists to experiment on themselves, right?
Yeah, right, exactly.
Cut this muscle or stimulate that.
Other questions? Okay, last question there.
Do you think it is needed to implant something? Because I was last time to, I think it's a place here in Barcelona. It's called Star Lab, where they sell a cap, which is like you can record and also you can stimulate, and of course it makes a non-specific stimulation because it just injects current and
through the scalp you get
through the scalp. and they show that they are using this to improve motor performance, and even they tried it on what was it? It was a person was in Guinness for doing some mathematical calculation rapidly. They trained it with the cap, and he got better, and the thing was persisting. So I was quite.
That's interesting. Yeah, you have to tell me about this. I'm not aware of it, but I want to know all about it.
What's interesting about that, you could consider, of course, that you only capture your signals on the brain, you send it off wirelessly, and then all your computation offline. This is a bit what you were after, I guess, no? Or you want to train the monkey to become a mathematician?
No,
well, some mathematicians are like monkeys, I guess.
No, I was thinking. I mean, when I saw that, I was. Sounds strange to me that such a non-specific stimulation could help actually. But so I will know.
Well, I want to hear. So right after this, you have to give me the website.
But let's be careful. There's also quite some propaganda behind these kinds of things, right? Like lots of bikini girls with sort of headsets, like really very sexy. So, other questions?
But your point about the wireless loop is interesting because obviously you can do much more computation if you do it wirelessly. The problem is you get a much longer loop time. Wow. So you couldn't do if you go through a computer and you have the computer detect the pattern and then deliver the stimulation.
Some gigahertz drop band.
Okay, maybe.
I think it's not a big deal, you know?
Yeah.
Okay, so last question by Giovanni.
Yeah,
you cannot get a prosthetic device. Talk to me about it.
Okay, yeah, thank you very much for the talk.
Thank you.
So I have a question and a comment. So, well, the question is, with your Hebbian plasticity and conditioning paradigm, so how far you think you can go? So if you can really teach skills in this way, or
no, these Hebbian plasticity effects are in specific circuits, but you could probably think about ways to enhance skills by having more complicated contingencies that are implemented with this. You'd have to have a more sophisticated computational capability, but yeah, I think you could.
Well, the comment is related to this in this plasticity. In the plasticity slide you have shown also the peaks of activation, they decrease over time.
Yes.
Still, so the problem is, if this plasticity is also linked to a behavior, would also the behavior decrease, so the behavior accuracy decrease over time, or would it be conserved? Because
yeah, so that's a good question. The types of changes I've shown you aren't big enough to show up in behavior. For example, this corticospinal change is at the level of a single cell. Well, actually, other cells that are synchronized with that cell also change their effects. But to detect the effect of that on behavior would require really subtle detection strategies, which I'm skeptical would be worth the effort.
Well, because in a sense, if plasticity, if the peaks of activation decrease, but the behavior is conserved, then it will be some efficient coding. So in a sense, you just decrease the activation because they code more efficiently, still the behavior is preserved. And there is a lot of, there are also a few theories on how, at some point, because of Hebbian plasticity, you grow up very strong links, and that's very inefficient for the point of view of energy. And then, from time to time, you should decorrelate everything a little bit, so that you code still the same behavior, but much more efficiently. Yes. So this theory will explain why there is this decrease in activation.
Yes.
But still, it only makes sense if the behavior is preserved in a sense. So this efficient coding theory is for, also assumes that you are not losing the behavior, otherwise it's... So this is why I asked you the relations between this Hebbian plasticity and behavior, but...
Yes. So I think that, strength training and so forth changes the strength of these connections and transfers into other behaviors. That's true, but you have to think a little more about how to make that, detect that change behaviorally.
Well, I always talk to Tony when I need to decorrelate my brain. So, Eberhard, thank you very much for a great talk.